The overall goal of this procedure is to generate viable cell-laden constructs with a complex geometry using a 3D bioprinter. This is accomplished by first isolating human adipose tissue stromal cells from culture. Next, the cells are mixed with freshly prepared oxidized RGD conjugated alginate bioink. In the final step, the cell-laden biomaterial is extruded via bioprinting. Ultimately, confocal microscopy is used to analyze the viability, proliferation, and migration of the bioprinted cells.
The main advantage of this procedure over existing procedures such as scaffold forming and cell seeding is that with our procedure you can directly place the cells and cell aggregates exactly where they need to be to form the tissue. Demonstrating the procedure today will be Sarah Grace Dennis, a graduate student from my laboratory.
Begin by seeding 350,000 human adipose tissue stromal cells onto a treated T75 flask in 15 milliliters of low-glucose DMEM for expansion in a cell culture incubator. When the culture reaches 80% confluency, remove the medium and rinse the cells in 5 milliliters of DPBS without calcium and magnesium. Then incubate the cells in 5 milliliters of trypsin and DPBS for 2 minutes at 37 degrees Celsius. When the cells have detached, stop the enzymatic reaction with 3 milliliters of cell culture medium and transfer the cells into a 50 milliliter conical tube. Next, centrifuge the cells